Hello.
Hello.
Hi, how are you?
Yeah. I asked you for a paper. I'm sorry I'm bugging you about it. It just, I managed to lose it both in paper format and on the computer. This is
Oh, no.
bad.
So did it ever really exist, or?
Oh, yeah. No, you commented on it a couple of weeks ago. It's energy wells and the difference between columnar and squamish cells. They were a least energy, or they filled a least energy well. And it's interesting. I don't think I want to include it, though, in my paper on second thought, because I don't quite understand it, and that's why it's intriguing me. But I don't have that for my committee to ask about, so I'm just going to kind of take it out of there.
Yeah.
I still want the paper somewhere, but not for that.
I don't want to be examined on it.
No.
Yeah. I don't know. I couldn't seem to find it. I didn't know what was it in this meeting that you saw it, or was
No,
it
I
something?
sent it to you about 3 weeks ago or so.
Yeah, I couldn't find the
because it's
email.
interesting.
Yeah. So I was looking for the email and I couldn't find it, so.
Yeah, it seems to have disappeared from my end too.
Huh. I don't know.
So you erased it, did you? I don't know. Anyway, it's, yeah, anyway. Dick sent me some papers that were more to the point.
Okay. All right.
One's called Hexanemic Crossover in Epithelial Monolayers Depends on Cell Adhesion and Cell Density. So that fits in with my Appendix C that I'm putting together.
Oh, yeah.
It's basically how cells are not, they're sort of hexagons, but they change shape, especially if there's forces on them, and then they become elongated, and then they're more like anemic crystal, or they behave like anemic crystal.
Yeah.
And it's just, it's easy to make them look like a crystal. I have see my mess behind me. Anyway, it's this thing.
Oh yeah,
this thing, and if you pull it from two of its strings, just two, you get I don't know if you can see the top there, but the top becomes elongated.
Yeah, you pull on it.
It's an elongated structure.
Yeah.
Especially if you pull it from opposite sides, then it's very elongated.
Right.
It's no longer hexagon.
That's awesome, Susan. I thought I was the only one who made models like that.
Well, this is made with a used box from Staples, which I'm trying to paint.
Sure. Yeah. I can't remember exactly what my friend Simon used to make his tensoricity, but it's something like that.
Yeah, well, that's sort of basically what I was doing, was making a tensegrity. But yeah, and then I went, it's the shapes, like they change all over the place. And, oh no, what's going on? Well, it's forces in the fact that the cells contract the sides on things. It's the combination. They can change shape easily.
Yeah.
Sometimes permanently, yeah. Just anyway, yeah. So that's Appendix C for what I'm doing. Yeah. Why are the cells hexagons? Well, they're not exactly, but try to get my trust model to do anything like that. It's like it thinks it's building a bridge. And if he doesn't put forces on the side and change it around, it's going, it's unstable. Well, yeah, it has to be. It's just,
Right.
yeah. Anyway, yeah, I've been having issues with console because it wants to build a bridge, and I want to build something that's somewhat unstable. So,
Yeah.
So in the end, I've just gone with, this makes it more unstable than that.
Right.
Or this is stable if you put a proper tensegrity in the middle of the cells, like a trijunction. If you put a stable tensegrity in there, then it becomes stable.
Right.
So that's what I wrote up, because that's what I was getting. And it kind of behaves like cells do, because you put a stable trijunction in, and it becomes stable. If you have sort of an unstable trijunction in, you can try various other things that kind of stabilize it, but the trijunction is really important. It's sort of the conclusion.
Yeah.
So yeah, that was good. I've got lots of insights about cells for the engineers I'm working with, and maybe that's the whole point, because they're going, I know nothing about biology. We are engineers, Susan. We don't know anything about biology. And I'm going, I have a minor in biology. Some people might call it a major.
Yeah.
I have some background in biology.
Yeah.
Not as much as I should have, because I hate biochemistry, but that's just, I don't know.
Right.
So I did write this thing up. It's 25 pages long and 8,000 words.
Wow.
But they were saying that I hadn't done any work, so that's written it all up. It's going to be 30 pages and 10,000 words. That's the goal.
Yeah.
Complain at me, and that's what you can have to read. I've been calling it Revenge of the Nerds. Anyway, I hope that he seems to he likes it, so it's good. I'm summarizing things.
Yeah.
So, yeah. How do cells behave?
Yeah. That's a good question, I guess.
Well, nobody knows, but
Right.
you can say it does some things.
Yeah.
So my tensegrity model isn't as good as I would like, but I think I should put it into a modeling system that does tensegrity, so it'll expect the thing to move. And it does. It moves all of a sudden. Like you have this here, and you put force on it, and it goes boop like this, and then again. I think it has some, just depending on the build, it has some rather sharp moves, which are called instabilities in it. Every once in a while, you get an instability.
Yeah. Yeah.
Yeah. So I don't know.
Well, that's good. Thanks for the update. I'll actually have a paper on that. It just came out. And we'll talk about that later. But remember we talked a long time ago about liquid crystals and about the Desjardins book where he talks about like biology is liquid crystals and how cells are arranged like crystals, and it kind of goes into like the soft active materials area. Yeah.
So that's what that was about. That paper says.
I've certainly seen. I'm sorry.
Sorry.
So I was thinking about clathrates, and how clathrates are definitely more structural than free form. That the idea that inside cells is a lot. The physics is a lot different than we perhaps suppose. But I'd be really interested to see that. Really.
Yeah.
You've got a link.
Yeah, well, we'll get to it. Yeah, I'll have the links.
I'll maybe get the references here that Dick sent me if I can, or at least maybe I should just give you the titles.
Yeah, you can put them in the chat.
Usually look things up with the titles.
Right.
It's amazing, but that's true.
So yeah, Morgan, how are you?
Good, thank you. Yeah, sorry, I'm doing like school run stuff, so mute me if
okay,
if I forget to mute myself. But yeah, good. I just posted a paper that I thought was very CompCell 3D or so. What's that package?
CompuCell 3D, yeah.
Yeah, CompCell 3D. Interesting modeling. And I was wondering though if these are both from Complexity Digest, whether you've covered that PNAS paper that it's like evolving systems. Oh well, it's funny because this is the first paper that my dad sent me.
Oh, okay.
And he's like, "Hey, is this relevant?" Which I loved. Yeah, I'll find the link here.
Okay. Yeah. So yeah, that's good. Yeah, let's take a look at this paper. Oh, this is the one, Epithelial multi-scale active liquid crystals. Actually, that's the paper I'm gonna talk about.
Okay.
But yeah, that's one of them, and that's brand new. And then this one is the BMC biology. This is the one that let's see. Let me share my screen here. This is the one that Morgan shared in the chat. Okay. Here we go. Right here. Is this it? Spatial biology of icing like synthetic genetic networks. Yeah, this is the one, I think, because I remember seeing this in the...
muted. Yes, definitely.
Yeah.
I'm just seeing now from Complexity Digest that David Krakauer also has a paper on machine learning and evolution.
Okay.
That sounds relevant to.
Yeah, I didn't see that one, but I saw this one come up. But yeah, this one is... So this is spatial biology, which means they usually consider different parts, different locations in the biological structure and how they're heterogeneous. Ising synthetic genetic networks. So there's this Ising model that we can use to characterize, and they're actually doing this in an organism. It's an E. coli 2D colonies. So this is synthetic biology where they're using a bacterial model. And they're building networks. Yeah. So yeah, what they're trying to do here is understand how spatial patterns of gene expression emerge from the interaction of individual gene networks. So you might have in a bacterial culture, you might have a pattern like a spiral pattern, and the cells are expressing things differently depending on where they are on the colony or in the culture. And so the question is how do they do that? How do they coordinate their gene expression or their gene regulatory networks to do that? So they've developed a synthetic experimental system. So you have an E. coli, you engineer a circuit, you let it, grow in a substrate, and then you can maybe modify the pattern. So they have a common theoretical framework for this. This captures the emergence of short and long range spatial correlations, which is where you take a single cell and you look at the neighbors out to a certain distance, and you look for spatial correlations, which are basically it's like time series, but you're doing it over space instead of time. So you're doing from T0 to T whatever, and then you're looking from T0 to T whatever is space 0 to space whatever in a certain direction, and then you're looking at the state of things intermediate to that. You get a spatial signal that resembles a time series. And then you can look at how that correlates. There's a correlational structure to that. So you have these interacting gene networks, so each cell has its own network, and it's being differentially expressed, and so that's what they're trying to do here in this paper. And then, this is guided by the combined mechanisms of the contact process lattice simulation. So they use a lattice simulation and then a two-dimensional Ising model, which they call CPIM. They describe the spatial behavior of bistable and chemically coupled networks. So the cells are chemically coupled, they're signaling each other. They're also bistable, they have different stable states. So the output can be like maybe, one color or another color. You can do this with like in a toy model where you have, distinct colors that can be achieved. It's just that there's this, this chemical influence between the cells. And so it's either synchronized with the neighboring cell or it's distinct, and you get a different state coming out of that. Self-organize into patterns of long-range correlations with power law scaling. So that means that they're these long-range correlations, and you get power law scaling, which is the long tail, and short-range anti-correlations. So there are differences at different scales. If you move out from the cell to farther out, you get these long-range correlations. In the short term, you get anti-correlations, and that's kind of like in a Turing morphogenesis model where you get a boundary where the cells are different states.
Yeah.
Then the Ising model is it's a model of ferromagnetic activity. So they've used this in physics this is a traditional model in physics to model complex systems and systems of interaction. So usually they're looking at the spin of the single cell or the single unit in the model, and then they have this network of units that will form this Ising model. And so that's what they're doing here. And then they, kind of go through, they talk about their two-dimensional Ising model and growing cell populations. They call it the contact process Ising model, which is this specific type of instance of the Ising model.
So it wasn't cellular pots. Okay.
Oh, you thought it was cellular pots, or?
No, I just when I hear the CP, I've got CompuCell 3D in my head.
Yeah. Yeah, the CompuCell 3D uses cellular pots, which I don't know how different it is from a Ising model. I'm
curious. Yeah.
Oh, yeah. Is it? Do you know what is your opinion on that?
No, not sure, but Ising is a generalization of pots, or like vice versa.
Yeah.
So yeah, curious about that.
Yeah. Okay, so then they talk about we implement this in a growing population of cells. We generated a lattice model that combined the contact process, which is citation 36, which is this paper from 1974, contact interactions on a lattice. And then, this is where you have a two-dimensional lattice, and the cells are interacting, and there's this contact process. And it's a formal model in physics, and actually in probability, representing cell population dynamics with the two-dimensional Ising model using the Metropolis algorithm, which is like a distance equation. So equation of state calculations by fast computing machines from 1953. So this is like the original one of the original Metropolis algorithm citations. And so that's their model. So this is where it's an interacting particle system that follows colonization, extinction, and differentiation dynamics of particles on a two-dimensional lattice of end sites. And so you have these sites that are unoccupied or occupied. You have different cell types that come into different regions, or they go extinct, or they differentiate and they spread into new areas. And you can model all those dynamics in this model. So as in the original Ising model, cells are fixed in their position. It can only interact with their nearest four neighbors with interaction energy H, which in the absence of external perturbations is determined by this Hamiltonian. So you have these Hamiltonian dynamics in what is essentially like a cellular automata or a cellular POTS type model. And then they have this this parameter here, which corresponds to the state of differentiated cells. Magenta, which is +1, or green -1. So this has two states, and then the IJ, which is this term here, indicates that the sum is only between neighboring pairs of cells. So they're only allowing for short-range interactions for I, and then J represents the contribution of free energy made by the coupling interaction between these pair of cells. So I is the distance and J is the energetic aspect. And so it's this pair of terms. And then we assume that J equals +1 for ferromagnetic systems and J equals -1 for antiferromagnetic systems. This implies that the interaction energy between neighboring cells is minimized when they have the same state in a ferromagnetic system or the opposite state in an antiferromagnetic system. So they basically use that as a proxy for polarized states. So they have these two states, the two different colors, which are the, I can't remember the colors they're using now, magenta and green, I think. And then they're using that as like the magnetic, ferromagnetic and antiferromagnetic, just to polarize the two. So when you flip it to the other state, they're different colors. If they're in the same state, they're the same color. And then they kind of go through the physics here. Then they talk about the synthetic gene networks of spin-like behavior. So the spin comes from this ferromagnetic aspect. And what they do is they apply that to this synthetic gene network where they create an M-cherry reporter or a red fluorescent protein or RFP reporter and a green fluorescent protein reporter to report the state of these networks. So you get the red and the green, and they're switching back and forth, and that's where we're getting our colors. So basically, that model applies to this gene network. The output of the gene network is this reporter expression, which is either red, magenta, or green. And so the SGN, or the synthetic gene network, consists of this reporter, a state reporter, a switch module, which has two repressors, LAC1 and TEDR, that repress expression of each other. So it's mutual repression, allowing cells to adopt only one of the two possible states at any one time. Then 3 is a coupling module, which allows the production of one of the two coupling signals for each state. So there's this LUX1, something synthesized from LUX1 from Vibrio, and then this H12 HSL, which is synthesized from LAS1 from Pseudomonas. So they've used these different coupling signals from nature to do this, build the circuit. And then they have they put this packages into two vectors, a reporter vector and the ferromagnetic or antiferromagnetic vector. So that's the way they do this. So basically this one is determining the state and this one is determining the reporter. So they have to put in the reporter and then they have to put in the gene network or the circuit. So yeah, this is a pretty cool paper. And then I don't know if they have any pictures in here of different things, but here's some of these. So this is an example here of the patterns that you get. So in figure 3, you have the antiferromagnetic here, the ferromagnetic. These are the two reporter vectors. So vector 1 is here, where you have, it's probably going to pull it up now. Okay. This is where you get the patchiness between green and magenta. This is where you only have magenta. So that's vector 1, antiferromagnetic. For vector 2, of course, you have the green, and that's here, and I think you have a little bit of magenta in here. You get, for ferromagnetic, you get this mixed population where you get strong green and magenta expression across the surface, and you get these sort of this patchiness that you might expect from, say, like if you've ever seen a BZ reaction. It kind of looks like that, but it's these are all bacterial cells carrying these gene circuits with these reporters in them. And so you can see that the CPIM simulation is here, where they try to recapitulate these two states. So here it's mostly gray cells with some magenta and some green. Here it's all magenta and green, and they have this pattern formation. So the ferromagnetic state of this system gives you this bistable sort of pattern formation in this colony. So you can see here. So that's that paper. And then, so this is more CompuCell 3D type modeling than finite element modeling. Yeah, it's and then Susan gave this these two citations: Epithelia or multi-scale active liquid crystals, which we'll get to, and then hexanematic crossover in epithelial monolayers depends on cell adhesion and cell density. So we talked about nematic organization of cells when we talked about our liquid crystal biology stuff. So that's and then this is the hexagonal organization of the lattice. That's what that's referring to. So yeah, thank you for those references, Susan. I guess Dick got the references for you.
Yeah, it's from Dick.
Okay.
Yeah, he was helping me out for my appendix C.
Oh yeah.
Of my paper. I don't know. He probably didn't know that, but it's it helps tie it together with some of the other things I've been seeing. So I thought I would put it together. This is why cells are hexagon-ish. Yeah.
So this is something else Morgan found, Frontiers paper on. This is the paper Unifying Complexity Science and Machine Learning. So this is kind of a sort of more of a theoretical paper where he's kind of bringing together complexity science and machine learning. Yeah, go ahead.
I just saw it, but it kind of reminded me of a Richard Watson kind of title.
Yeah.
Yeah.
So this is whereas complexity science represents a coarse-grained paradigm of understanding, machine learning is a fine-grained paradigm of prediction. Both approaches seek to solve the Wigner reversal or the unreasonable effectiveness of mathematics in the adaptive domain, where broken symmetries and broken ergodicity dominate. This is where we get this complex behavior, get symmetry breaking, we get ergodicity breaking, or we get the breakdown of like predictability in systems, and where systems explore different states differentially. And this is basically the heart of complexity. We might get into that a little bit in terms of complexities like symmetry and ergodicity at some point, but just suffice it to say that that's kind of one of the principles of complexity science. In order to integrate these paradigms, I introduced a meta-Occam, which is like Occam's razor, which is the simplest explanation is the best, except it's a meta rule that he made up, which first of all moves minimally from the description of a model. Yeah. So it moves minimally from the description of a model to a phenomenon, for a phenomenon to a description of a process for generating a model. So this is like Occam's razor for sort of this description to a process of generating a model to approximate the thing that you're looking at in nature. And then 2 describes low-dimensional features or schema. And so then he talks about reinforcement learning and natural selection, which are both parsimonious in this revised sense of the minimal process. So this is where he gets into, reinforcement learning and natural selection as being kind of connected in that way. So it's really good. Thank you. Oh, do you have anything more you want to say about that, Morgan? Or?
It was more the next paper that I had seen, but I'm still thinking a lot about the Saturday morning meeting in terms of introduction complexity science
Okay.
and good ways into them. And I always found that the Richard Watson work was at least good for kind of data scientists. If they saw evolution as a kind of a learning process, that it kind of helped them into biology. You know?
Yeah.
So if we could do the same for complexity, that'd be awesome. Complexity science, that'd be great.
Yeah, I think so. So this PNAS paper is on the roles of function and selection in evolving systems. This is, I think, I don't know who these authors are, but this is where another one of these grand scheme papers.
I recognize them. Yeah.
So this is where the universe is replete with complex evolving systems, but the existing macroscopic physical laws do not seem to adequately describe these systems. Recognizing that the identification of conceptual equivalencies among disparate phenomena, and that means that you could use, say, something like a power law to describe systems is far-flung as like transportation networks or social dynamics in humans, or like cell behavior in cell biology, or even like things at the scale of galaxies or the universe. And so the thinking is that you can use these rules or these mathematical laws to sort of characterize this wide range of systems, and that's sort of the basis of complexity theory. Now, some people might argue that that's incorrect, but that's kind of what we do with statistics as well. We use statistics on any we just assume that any system that does things has a distribution, and we try to characterize the distribution, and then we have tests for that. So it's not, I think it's, it relies on sort of a universality, but it's probably a pretty good approximation of things. So we recognizing the identification of conceptual equivalencies, and then we approach a potential missing law by looking for equivalencies among evolving systems. We suggest that all evolving systems, including to but not limited to life, are composed of diverse components that can combine into configurational states. So you have these, collective structures that are configurational. They have, it may be like protein folding, or it could be like a cell colony where they have these different configurational states. So, if you want to know kind of the, what's possible in a system, you look at these configurational states and you say there are a lot of potential configurational states. Now, a system will settle into maybe a subset of those. So there might be one or two common ways a protein folds to make it functional, but there are a lot of potential states it could take. And in fact, it may fold into many of those that are not functional. And we need to be able to know kind of, usually the viable configurational states are usually a minimization of energy, or there are other historical contingent reasons why you get the subset of configurational states. But basically, that's what we're doing. We're taking this vast set of configurational states, which is usually any set of possible combinations or all set of possible combinations, and reducing them down to some viable set. And that's what a lot of complexity theory is, and a lot of algorithmics for that matter. So these configurational states are selected for or against based on function. We can then identify the fundamental sources of selection, which they propose as static persistence, dynamic persistence, and novelty generation. So novelty generation, of course, is where you get like new forms. Dynamic persistence is where things are persistent over time, and then static persistence is where they're static over like locally, not over time or over the course of evolution, but in the current state. So you could map this to like development, static persistence, evolution, dynamic persistence, and novelty generation, which would be like mutation or recombination. And so that's how you would think about that, maybe biologically. And then we can propose a time asymmetric law that states the functional information of a system will increase over time when subjected to selection for functions. So this is where we have this increase of information over time which is usually the opposite of the way we think of entropy, which is a decrease in information over time. We also think of when we reduce the number of possible configurations as a reduction of energy over time. But actually what they're saying is that information increases. So we don't have, whereas there's minimization of energy, a minimization of sort of configurational entropy, but there's an increase in information. And so we've been talking in separate conversations about the distinction between informational entropy and energetic entropy. And this actually fits into this because, you have this situation in, say, Maxwell's demon, where you have this, over time you get a decrease in energetic entropy, but you can have an increase in informational entropy. And Maxwell proposed this as like a demon that sorts these configurational states, because you can't say that there's no natural process for this, at least in Maxwell's time. So he proposed that there was some demon that was doing it, and this is just like an unexplained agent. But of course, it's not really agency; it's this sort of process. So yeah, we still don't really understand fully how this works, but we know that there are increases of information in these kind of systems. And so this is another good paper. In terms of explaining complexity theory, I would definitely try to. I'll try to bring together some resources, because I think that's there are some key concepts that need to be kind of unfolded before you get to methods. You can give people a box of methods, but there's some really strong ideas about things that are that need to be people need to understand those before they can start thinking about complexity. They're maybe counterintuitive, but it's it's something that like ergodicity. What does that mean? Or these sort of informational dynamics. And so we're doing that in another group, but we haven't really moved very far on that because we just the question kind of came up in the last few weeks. So, all right, Morgan. Well, thank you for those resources. Okay, so let me go back to the stuff that I was going to talk about with respect to liquid crystal biology that we've talked about this in past meetings. So there's been a Quanta magazine article, and then there's a paper associated with it in Nature that I wanted to talk about. So this is an image here of sort of a liquid crystal array in biology of cells, and this quote down here: before they can apply the predictive power of fluid dynamics to living tissues, so they're looking at fluid dynamics in living tissues, scientists must first identify the symmetries that equate a sheet of cells with liquid crystals. So what they're saying, what they're doing is they're using this liquid crystal theory, or the soft active materials theory, to describe sheets of cells. And so in the paper, they're going to talk about epithelial cells and some of those aspects in epithelial sheets. So this should be really relevant to what Susan's been talking about and writing about. So this is where this person here, Luca Giomi, still remembers a time when he watched 2 videos of droplets streaming from an inkjet printer. The videos were practically identical, except one wasn't a video at all, it was a simulation. You could predict everything about the ink droplets. So this is a highly predictive system. The simulation was powered by the mathematical laws of fluid dynamics. And so now you can actually use those mathematical laws to describe maybe cells in a sheet. And so this is this paper in Nature Physics they conclude that sheets of epithelial tissue act as like liquid crystals. And you define liquid crystals as materials that are ordered like solids but flow like liquids. So they have this order of a solid, like a sheet, and then a static sheet, but then they flow, they migrate like liquids. So they have this dual sort of aspect to it. To make that connection, the team demonstrated that 2 distinct symmetries coexist in epithelial tissue. These different symmetries, which determine how liquid crystals respond to physical forces, simply appear at different scales. So there's there's a solid and then a liquid state. And if you get to a certain scale, you get this sort of behavior. So in previous meetings, we've talked about jamming behaviors and jamming phase transitions. So jamming phase transitions are where you have cells in a pipe that are flowing in a pipe, where they're flowing in a medium, and if they're few enough cells, they can migrate freely. They can move around. They don't have any problem migrating or moving around. But at some point, there's a critical point at which the sheet jams. So you get this fluid behavior, and then the sheet jams when you get to a certain density, and then they sit in place. So you get these phase transitions are very quick. They turn on and turn off at a certain critical parameter, in this case, density of cells, and you get these two different states. So this is kind of what they're getting at with this. And so this insight could make it easier to apply the precision of fluid dynamic simulations to living tissues. And this person, Giomi, hopes to predict how human tissues move and deform during processes ranging from wound healing to cancer metastasis. So this is where you get a lot of different, systems that we talked about epithelial cells being involved in many different processes and in many different systems, and they're different in terms of their shape, in terms of the sheets that accumulate in these different systems. Sometimes they serve a protective function, sometimes they serve more of a functional role, but they have these very diverse sets of states, but they behave in a similar way. And so that's really interesting. So they talk about flow and symmetry. So liquid crystals flow like fluids, but they still have a degree of crystalline order, sort of inherent symmetry or directionality that is a bit like a grain of wood. So there's a certain grain of the wood. The wood plank is strongest along its grain, and such. Also, liquid crystal's response to stimuli depend on its symmetry and orientation. So there's a grain of the liquid crystal surface or the interface, the sheet. This directionality, called anisotropy, is the optical magic behind modern liquid crystal displays. That's why they talk about liquid crystal. It's the same physics. They just use it's in electronics instead of in biology. So they use liquid crystal displays. It has the same aspect. It just refracts differently depending on its orientation. So it's the orientation of the things inside of the collective. In liquid crystal displays, there are these little LEDs, and in biology, it's cells. So we can use this principle to describe tissues and their orientation, things like that. So this is where they talk about nematic orientation. So these efforts at a geometric roadblock. Experimentalists and theorists couldn't agree on the tissue symmetry. So this is important to know the orientation of the cells in this case, and it's key to this theory. So you have to be able to know the orientation to predict the fluid dynamic behaviors. In simulations of small groups of cells, theorists could describe tissues as liquid crystals with six-fold hexatic symmetry, which is the hexagon shape. And so there's the symmetry of being a hexagon and being in a tiling where, like Susan said, you have these threefold junctions, which are important they form sort of naturally when you have hexagons next to one another. And so this is where the stability of these sheets come into play. But also this orientation is important in the states that it achieves. So you can't have this, change in phase of the tissue without this sort of symmetrical geometry. So, but in experiments, tissues instead acted like fluids made of bar-shaped particles with twofold nematic symmetry. So this is where you have a nematic symmetry. It's kind of like where you have little uh, bars or little, little like if you see bacteria, kind of grains of bacteria. They're long and thin, and they orient themselves in that way. That's the kind of symmetry. So there's this nematic symmetry, which is the twofold symmetry, and then there's this hexatic symmetry, which is the sixfold symmetry. So if it's a bit like if you poured a barrel of toothpicks in a tube and watched them flow. That's what nematic symmetry is. Let me see if I can draw this out. This is actually an important concept. So if you have nematic symmetry, and this is what they talk about a lot of times in liquid crystal theory and soft active materials, you have these, and these are like bacterial cells that may be packed in a sheet. And this is nematic twofold symmetry I'm drawing. So you have these long, thin cells that look like bars that are kind of in a sheet. And they're packed densely here, so they're in a stable state. They're in a static state. And this is twofold symmetry. So this is where you have twofold symmetry. The twofold would be the right side and the left side, where you have this orientation towards in this direction or in this direction, and not laterally. So you have these left-hand sides and right-hand sides of the sheet. And so you also have a top and bottom, but flows might occur along this axis. So it's twofold, and you get that. Now, hexatic means you have this hexagon. Yeah, there we go. And then you have other hexagons kind of tiled next to it. So you have these hexagons here. Well, let me go to the next hexatic. And so this is sixfold symmetry. And so you have the sixfold being this side, this side, this side, this side, this side, this side, and you have that for every cell. And so the thing is it could move in these directions, but there are other cells blocking it, so you can't necessarily, you're jammed. You can't move in that direction. You're stuck there. Now, all the cells are stuck there, but they also all have this hexatic symmetry, so the stability of the sheet is intense. And so this is where you have this trijunction, like Susan was saying, that this is a very popular term in the literature now, where you get these three cells that come together at this hexagonal, or at this point in the hexatic symmetry. And so this is important for stability because hexagons are actually quite stable, and having a sheet of hexagons, like beehives, it's hexagonal, and they store honey in the hexagons. So this is like
it's a stable tiling.
Stable tiling, yeah.
Yeah, and like I say, cells do move, so it changes them, and if they get too elongated, then I swear the top is moving separate from the base, like the apical part of epithelial cells is moving separate from its base. And if you get too much of a force on the top of it, it'll move the bottom. That's my theory, and I was hoping there might be some literature that says that, because it makes sense.
Yeah. So it would be like this hexatic tiling becomes sort of a nematic tiling, or?
Yeah, it becomes elongated, like that, and then it moves after that happens. Like it'll turn into, well, you can, if you get rid of the trijunctions, the cells become a mobile mesenchyme type cell instead of epithelial cell, and there's a very small genetic switch there between the two of them. I was reading about it, and so they're related. They're like they become, it's a epithelium mesenchyme transition, and it happens all the time. So it has something to do with this transition.
Oh, it's interesting. Yeah.
Yeah, it's interesting. I don't know where. I wish that there were more images of both the top and bottom of the cell, the apical and basal part of epithelial cells. There are some papers with them, with that, but I'd like to see it. When they're not aligned, they're not straight up and down. You can get some microtubules in there stabilizing the sides of the cell as well. Like there's some conditions where the sides are stabilized, and the trijunction and the basement membrane kind of work together too to stabilize the cell. So I don't know. It's all there's a lot going on. It's hard to tell.
Yeah. So if we look at this, if we had like a hexagonal cell, something like this, then what you're saying is that this is the apical end here, and then this, draw the side view, which would be like this.
Yeah, there's a side view.
All right.
Yeah. So what I'm saying is the top there moves separately from the bottom and can become elongated, and then that puts pressure on the basement membrane, basal part. Yeah.
So the apical would move up like this, or be forced up.
That's when it dies. Yeah, it gets forced up and it gets squished out of there when it dies.
Yeah.
Like it can be ejected from tissue and it moves up like that. But it moves to the side, like you have in the first drawing there, the apical part, so that it's elongated.
Okay.
The top apical part can become elongated, and then the sides are at a tilt.
Yeah.
Yeah.
Yeah, I see what you mean. Yeah.
So if you pull on the apical part of the cells, it eventually puts pressure on the basal part of the cell. And I think they move separately, and after the apical part gets stretched enough, it'll move the cell. Like they're attached, but they'll become detached if there's enough force on them.
Yeah.
And they're a confluent tissue, so they kind of change partners instead of if they're anyway, since they're confluent tissue, they tend to change partners.
Right.
Until they turn into a mesenchyme cell and just the whole sheet moves off somewhere.
Yeah.
Becomes a crawling cell. I have collected a bunch of images, sort of built on I called it some cell biology. I have a bunch of images. It's interesting.
That's great.
And some of when an embryo is hatching out of its zona pellucida, apparently it loses its adherence junctions. Okay. So it loses some of the junctions that are holding the cells together, and it becomes more fluid. It can go through a very tiny pore. So it's also prevalent in cancer and in wound healing, and kind of across the board thing.
Yeah. Okay. Well, when I go to the paper, which is the main, this is Nature Physics. So this is epithelial or multi-scale active liquid crystals. So the word multi-scale is interesting because you've been talking about active liquid crystals, but like there's this multi-scale aspect as well. So this is where we're talking about embryogenesis, wound healing, and cancer progression. They rely on the ability of epithelial cells.
Yeah, there we go.
Yeah.
It summarizes it for me. but it doesn't say whether the top moves different than the bottom of the cell. Anyway, it's annoying.
So it relies on the ability of epithelial cells to coordinate their mechanical activity over length scales that are orders of magnitude larger than the typical cellular size. So if we go back to our hexatic sheet, we have this, the sheet that expands outward many cells wide, and then, this whole sheet will behave a certain way as opposed to just the single cell. So that's what they're getting at. And then, we know that signaling pathways are involved, but also there's this physical coordination and this liquid crystal order, which is this long-range order, is a prominent example. They say prior studies have given inconsistent results, whereas nematic order is often invoked in the interpretation of experimental data. Computational models have instead suggested that hexatic order could serve as a linchpin for collective migration in constituent cell layers. So this is 6, 7, and 8, which are these references here. Role of cell deformability in two-dimensional biological tissues.
I need to get those.
Yeah.
Sorry.
Thermally driven order-disorder transition in two-dimensional soft cellular systems. And then 8 is hexatic phase in a model of active biological tissues, and that's in soft matter. Yeah, I'll send you this paper and I can send you this stuff. So in this letter, we've resolved the dilemma using a combination of in vitro experiments, numerical simulations, and analytical work. We demonstrate that both nematic and hexatic order are present in epithelial layers, with the former being dominant at large length scales. So the nematic order is dominant at large length scales, and the latter at small length scales. So you basically get this sort of hexatic order in a smaller space or a smaller sheet. But then in larger sheets or longer order, you get this nematic order, which is just this twofold symmetry. And so I guess what they mean is that over long spatial scales, this hexatic symmetry breaks down or something, but it retains a twofold symmetry.
Okay, I'm not sure what they mean by that.
Yeah, I don't really. Yeah, I'd have to look at like something of an image
Yeah,
to see.
I need to read this.
Yeah.
But it's Appendix C, so it has to wait for Appendix B to be done first.
So this is where you have, yeah. So you have this is hexatic order here. You have these different patterns of cells that are all packed in as hexatic arrays. Then you have this, they call it the winding number, which is just a description of the order here. And you see the different ways that these can be tiled together. They show kind of so they segment a bunch of cells and they put this model together. And then they show the symmetry here of sixfold symmetry at the corners. So you have these vertices which serve as the points of symmetry, and then they can be the junctions as well. So that's figure 1. Let's see, this actually shows, this is the same thing where you get this hexatic order in a model. This is the symmetry in MDCK cells. So the MDCK cells, the type of cells they're using here. So these are human cells. So you get this distribution here with a long tail in one case, and in the other case it's this sort of normal distribution. What is B? B is all about where did they say B here? I'm not seeing B listed. I think they missed it.
Oh, great.
Yeah, I don't know what the B. I don't know what B is, like what these two distributions are. But this is, one is a power law and one isn't. So it may be like the static and dynamic, but they describe it more in here. So that's just about the cell distributions and the packing. Then figure C is where you have this coarse graining in multi-scale order. So this is where you're getting this transition from hexatic to nematic. This kind of shows the, so this is coarse graining, where they just kind of coarse grain the model, and they kind of put this together. Let's see if they have a better image of this transition between the different types of symmetry. I don't think they do, actually. They usually in these papers have a lot of supplemental figures and things, so this may be also where it is. Let's see, there's extended data figure 1, misidentification of nematic defects in honeycomb lattice. Number 2 is probability distribution of cell coordination number. So you have these different models here, Voronoi model, MPF, and the experimental observation. It just shows the other, the measure coordination. Okay, these are the probability distributions of the order parameter for different. So the distributions we saw were for the Voronoi and MPF models. So this is orientation at different length scales. Let's see if this has a better idea of what they're talking about here. Okay, so this is it here. Nematic and hexatic shape parameters at different values of the coarse graining radius R. So you take a point, you draw a radius out, and you say how far out can we go. The scale here, the scale bar is 10 microns. So this yellow, so it's basically like between 3 cells, it's 10 microns. So that gives you an idea of the scale here. You have this orientation. You can see that the orientation changes over long distances. So, you have these sheets, but they change their orientation gradually. So these are the nematic bars here. This is a force field that shows sort of the orientation. And you can see here that you have changes in orientation of this. I guess this is a trijunction or something similar, where it's changing its.
If a cell dies, it causes a disturbance in the area, and you get rosettes. Like you'll get a cell orientation that kind of goes in a circle.
Oh, okay.
Like that's basically all I know about that. But.
Yeah. Let's see. So they talk about in both top and bottom panels, positive and negative defects are marked in red and blue, respectively. So these are the defects. Okay. +1/2 for nematic and +1/6 for hexatic, because we have the twofold versus the sixfold symmetry. Spatial resolution of the shape parameter is the same on both panels, but the number of orientation markers in panel B, which is here on the bottom, is half of that used in panel A, which is on the top for the sake of clarity. So this is kind of where they're showing this, the defects, and then this order, which is here. So this is I guess the clearest they make this. Is this helpful, Susan? Or?
Yeah, I need to look at this. I've been told that I'm supposed to reduce my model to something. Well, that's reduced. There you go. Somebody did it already. I don't have to do it.
Yeah.
Yeah.
So there are a lot of references in here and a lot of methods that we're not going to go over, but it's definitely very physicsy. You have topological defects are characterized. There's a correlation, spatial correlation function, and then they're also doing their multi-phase model, which is the MP model, or the MFP model, and then you have the Voronoi model, which is of course the Voronoi, based on the Voronoi tessellation, and the dimensionless numbers, which are these measures that they were using in the paper. So there's a lot of stuff here to put into the pot.
That P number.
Which one?
P number in the Voronoi model.
Oh, yeah.
It's just how elongated the cell is.
Okay. Yeah,
this one here. If it gets elongated enough, then it's mobile. Then it's acts like a fluid.
Okay.
Which that's why I said think it's the top gets elongated, and then it causes the cell to move because it's putting too much force on the basal part of the cell that's attached, so it becomes detached and it moves.
Yeah.
Maybe. I don't know. Does somebody say this? Do they look at both the apical and basal part of the cell? My question.
Yeah, I don't think so.
Yeah. Anyway, I need an appendix C. This is all really good stuff for it. So
yeah.
Good enough. It's good enough.
We've talked in the past, like a long time ago in the meetings, about organoid models and using them as a way to like characterize cell segmentation and things like that. Yeah, it's kind of interesting. Well, it's not surprising they bring up a organoid model here, but it's kind of nice to see that they're working this out in terms of sort of a state of a sheet of cells. So, yeah.
Yeah, it's nice to see them combining things.
Yeah.
Because they should. They should be giving similar results.
Yeah.
They should be connected in some way. Because both characterize the way cells move. So why wouldn't both theories combine?
Right.
Like that? Should be a connection.
Yeah, yeah. Well, Morgan, did you have anything more to say or anything to add?
So,
He might be busy right now.
oh.
Well, anyways, yeah, thanks for the papers, Morgan. It was great to look at those and consider, and I think that helped us later in the meeting understand, like, some of these physical aspects of cells, and, they don't use, well, they use some of the same terms, but I think like, we've been talking about putting together some things of how to help people understand complexity theory. And I think that's maybe a key aspect of this work too, is that, you have to understand like some of the principles of complexity to really understand what's going on in some of this work on cell, like phase transitions and cell sheets and the outputs of genetic regulatory networks and things like that. So
That should be a next study for myself, is complexity theory, so I can understand it.
Well, yeah. But yeah. Yeah. So,
oh well.
Yeah.
Oh, okay.
Well,
anyway,
okay.
I'm still working on this, and I suspect they are going to assign me more things to do on Thursday.
Yeah.
But we really like your paper. We want to discuss it. Yeah, you're gonna go into it in detail and say we want more on this aspect of it. Yeah.
Yeah.
You see it happening.
Yeah.
So, okay.
Well, yeah. Good luck on that.
Thank you.
And thanks for attending again, Morgan. And talk to you next week.
Yeah. Thanks again.
All right.
Cheers.
Okay, thanks. Bye. So now I'd like to go over a paper on configurational space and proteins. And so this paper is called The Genetic Architecture of Protein Stability. And this paper considers the configurational space of protein sequences and protein folding, and how it's very large, but maybe we can reduce this using the genetic architecture to maybe predict viable configurations or things in that neighborhood, so it can reduce the search space of, say, like a deep neural network trying to sort of solve protein folding problems. So the abstract reads, there are more ways to synthesize a 100 amino acid protein, which is actually 20 to the 100th number of combinations. And that's a lot of combinations. There are more ways to do that than there are atoms in the universe. Only a minuscule fraction of such a vast sequence space can ever be experimentally or computationally surveyed. So we have a problem of computation and a problem of experiment, where There's so many combinations, we can't possibly sort through all of them to find the viable ones. So we have this configurational space, and we have a high configurational entropy of that space. And we want to reduce it to things that are viable, to something that's tractable computationally. So this is why we're interested in sort of these alternate methods. Now we've used deep neural networks to navigate these high dimensional sequence spaces. This is a tool that gives us a lot more power than, say, like just using standard off-the-shelf algorithms, and certainly more than doing this experimentally, where you would have to go through one at a time and do these sort of combinatorial experiments where you do a large set of experiments in parallel. However, these models are extremely complicated and provide little insight into the fundamental genetic architecture of proteins. And this is in reference to deep neural networks. Basically, deep neural networks have a problem of interpretability, especially in terms of this problem. So we have this complicated algorithm, this complicated model that will spit out answers, but it doesn't give us any real insight as to how it arrived at that answer. And so, the idea is that there's some genetic architecture that leads to the production of proteins, that leads to the production of these amino acids. And so we can get these stable configurations, but we don't know what they're composed of or why they're composed of what they're composed of. So you can answer basically basic protein folding questions, basic biophysics questions, but you can't necessarily frame them in the context of genetics or of evolution. Here, by experimentally exploring sequence spaces that are larger than 10 to the 10th, which is quite a large sequence space, we show that the genetic architecture of at least some proteins is remarkably simple. So we're talking about this very large sequence space. So a lot of sequences are generated in the sequence space, a lot of possible sequences. But then we show that the genetic architecture of at least some proteins is remarkably simple, allowing accurate genetic prediction in high-dimensional sequence spaces with fully interpretable biophysical models. So as opposed to deep learning models, biophysical models are much more interpretable, especially in terms of the underlying biology. We can have, you can have our cake and eat it too. These models capture the nonlinear relationships between free energies and phenotypes. So we want to know something about how you get to a certain phenotype. What is, we usually would like to minimize free energy in a model like this. But there's also this model of phenotypes, and phenotypes are produced by sort of these free energy states that are minimized generally, but not always at every step. So we want to capture the nonlinear relationships here, but otherwise consist of additive free energy changes with a small contribution from pairwise energetic couplings. So this is where we have these additive free energy changes at every step of the protein production, and then we have the small contribution from pairwise energetic coupling. So they use this type of model to explain this sort of nonlinear relationship. These energetic couplings are sparse and caused by structural contacts and backbone propagations. So they use these local energetic couplings, these pairwise couplings, to explain some of these nonlinearities. Our results suggest the artificial intelligence models may be vastly more complicated than the proteins that they are modeling, and that protein genetics is actually both simple and intelligible. So this is where we talk about the map from genes to proteins. We have this genetic space, we have this protein space. That space compared to what deep neural networks are producing, the predictions that they produce, are actually simpler, but also more intelligible in terms of the biology. So this is where they talk about massively parallel experiments, allow the effects of single amino acid changes in proteins to be comprehensively quantified. And so here they kind of describe this space, the number of ways to combine one mutation at 34 different sites in a protein as 2 to the 34th, which is roughly equivalent to 1 1.7 to 10 to the 10th. So this is on the order of scale of 10 to the 10th. And this is 2 to the power of 34 combinations. So you have this very large space. It's very large. It's very hard to deal with computationally. Experimental exploration of such a large number of genotypes is extremely challenging. So this is a lot of genotypes that you would have. You'd have one mutation at 34 sites, and then you have the interactions between those sites. So it's very hard, this is going to produce a large number of genotypes. And our, technology right now, we don't have the sort of computational power that's needed for this scale of inquiry. To this point, we've analyzed sequence spaces up to around 10 to the 6th. So there's this gap between 10 to the 6th and around 10 to the 10th that we need to fill. But moreover, combining random mutations in even moderate numbers nearly always results in unfolded proteins. So, for example, only 2 to 8% of 5 amino acid variants and less than 0.2% of 10 amino acid variants in a small protein domain are expected to be folded. So the folding aspect is a part of this. That's, folding is not the dominant behavior. It's only a small portion of the mutations that are produced in the genetic architecture translate into folding. So, this is something we need to predict from things that don't fold. And so this is something that we can't really get at just by looking at the gene sequence and just saying there's a mutation that must be a fold, or that must be some functional significance. We know that from other studies of what mutations do in terms of disease states as well. Sampling even tens of millions of random combinatorial genotypes in most proteins will therefore provide almost no information about genetic architecture. So we look at these genotypes, we look at the mutational landscape. Most of it is useless. So it doesn't really provide any information on its own. You have to structure it in some way to pull the information out. Unfortunately, we don't necessarily know what mutations translate to the function of the proteins. So it will not be useful for training and evaluating predictive models beyond testing the trivial prediction that most genotypes are unfolded. So this would be something that we already know. We can kind of intuit it. So we don't need a model to tell us this, but this is kind of where deep learning models go. They basically tell us this, and this is not an insight. So people mostly have used deep learning to explore these high-dimensional sequence spaces. Deep learning networks are generative, which means that they generate a lot of solutions. With millions of deep learning networks of millions of fitted parameters have proven successful for diverse prediction and protein design tasks. However, these models have extremely complicated and difficult to interpret architectures and alone provide little insight into the actual genetic architecture. So the genetic architecture is separate from the model architecture, both of which are hard to understand, and in particular the deep learning network and how it maps to that problem. So it could be that the extreme complexity of these models reflects an underlying extreme complexity of protein, genotype to phenotype landscapes. So this is where we have what we've called the G to P mapping in previous meetings, and that is what aspects of the genotype map to changes in the phenotype. So can we describe phenotypic diversity in terms of genotypic diversity? And if so, how does that map work? Now we know it's not a one-to-one map, so we have to figure out what that relationship actually is. Alternatively, the actual genetic architecture of proteins might be much simpler than is evidenced by these models. So it's possible that the genetic architecture is very simple, and then we're overthinking this in terms of like the actual functional aspects of it. Such simplicity is suggested by the success of quite simple statistical models in four-dimensional sequence spaces. That's citation 19. So if we go to citation 19, so that's citation 19. We'll get to that in a minute. And the use of Potts-Hamiltonian models that only consider positional conservation and pairwise covariation in multiple sequence alignments of protein families to predict protein structures and activity. And this is citations 20 through 22. So we talked about Potts-Hamiltonian models like we talked about earlier in the meeting, the Ising-Hamiltonian models. These are Potts-Hamiltonian models, which are very similar. And so you can actually use those kind of models to predict things in sequence alignments and things like that. So these kind of models, there's this idea in complexity theory that we can use these type of generalized models. We can take that analogy from physics and apply it to biology and solve a host of problems. So this is, again, the assumption they're making. Here we use an experimental design that enriches functional protein sequences to explore the fundamental genetic architecture of high-dimensional protein sequence spaces with over 30 dimensions and over 10 to the 10th genotypes. So this is this large space, over 30 dimensions. This is going to be a pretty big model. Despite the complexity of deep learning models, we find that protein architectures are remarkably simple, with additive biophysical models providing very good predictive performance. So we're just using a biophysical model. We're using a very simple protein architecture, and we're finding, promising answers. Quantifying the pairwise energetic couplings, which is what they mentioned previously, between mutations further increases predictive power, providing excellent performance in high-dimensional genotype spaces. So you don't need a super powerful model to get at this problem. These couplings are sparse and related to protein 3D structures. The genetic architecture of at least some proteins is therefore very simple, with additive energetics and a minor contribution from sparse pairwise structural couplings. It's important to note that they say at least some, which is at least some proteins, which is basically saying that they did this on a subset of proteins, but by no means do they want to imply that this is a general thing. Let's move on with the paper here. Okay, so in figure 1, this is an efficient strategy to explore high-dimensional protein sequence space. So they have this model of mutations. They characterize the mutations in protein sequences. They have a biophysical model which infers single amino acid substitution for energies of folding and binding. They use a greedy algorithm to select optimal multi-mutant combinations, preserving fold-in functions, and then they map this on, they do combinatorial mutagenesis on the protein structure itself. So they're actually able to do this, in quite a compact pipeline for what they're trying to do. So they're actually able to get, I guess they're also doing a PCA here as well. So a shallow double mutant library of protein variants was assayed by abundance PCA and binding PCA, in combination referred to as DDPCA. So they reduce the dimensionality, followed by biophysical modeling to infer single amino acid substitution-free energy changes of folding and binding. So they reduce the dimensionality of the abundance and binding factors in this combinatorial mutagenesis. Then they do this, build this biophysical model to infer the conditions under which these changes are functional. They use this model together with a greedy algorithm to select a set of single amino acid substitutions, which when combined will simultaneously maximize both the predicted abundance PCA and binding PCA growth rates, which preserves both fold and function. So they do some other things as well, but the point here is that these models are quite good, at least in terms of what they've tested, and they are maybe a lot better, superior to deep learning models in a number of ways. So actually, let's see, we have some results here. So accurate combinatorial genetic prediction with simple biophysical energy models. Surprisingly, our original biophysical model, which is what we saw in the last figure, trained on abundance and ligand binding selections, double DPCA, DDPCA, quantifying the effects of single and double amino acid mutants only, explains as much as half of the fitness variance in combinatorial multimutants, where the vast majority, 94%, included at least 13 amino acid substitutions in the wild-type sequence. The only trained parameters in the simple model are Gibbs free energy terms for the wild-type, which is one genotype, and the single A amino acid substitutions, which is the non-wild type, and a two-parameter affine transformation relating the fraction of folded molecules to the abundance PCA score. So that such a large proportion of phenotypic variance is explained by an additive biophysical model, so they don't consider epistasis or other gene-gene interactions. That is trained on genotypes that only contain one or two genetic changes, suggests that the energetic effects of mutations in proteins are largely context independent. So mutations don't rely on context of where the proteins are found. They're just in the energetic effects are independent of that. On the other hand, a linear model that implicitly assumes mutation effects combine additively at the phenotypic level of multimutants, trained on the same ddPCA data, performs significantly worse. The linear model also systematically underestimates the observed phenotypic effects of mutant combinations, and a consequence of not accounting for the scaling of mutational effects due to protein thermodynamics, or global epistasis. And so this is where we consider, we don't consider epistasis at all. We actually can, let's see, for example, introducing a destabilizing mutation in an already unfolded protein has no effect on the fraction of folded molecules, which is not captured by a linear model. These results demonstrate a key advantage of fitting biophysical models. Accounting for global epistasis improves the generalizability of predictions beyond the local neighborhood of the training data. So this one, in the first model here, they talk about no specific epistasis or interactions, and that just simply means that it's not specific to the context. So specific epistasis means like it could be some specific interaction that we expect in a gene regulatory network or in a circuit of genes that contribute to a protein. And that epistasis might change if you find the protein being, having one function versus another. In this case, they just take bulk epistasis and they consider it as part of the model. In the second model, they don't consider epistasis at all, and that's the difference between these two models. And so that's where it suffers in performance. So this is an example again of the input values. These are amino acid sequences, energetic terms, and then these are some. There's a summation here where those are put together. This is the unfolded state versus the folded state. There's an energetic term there, and we get our thermodynamic entropy here. We also, from this summation of the amino acid sequences that are encoded in these energetic terms, we get these trained parameters in the model, which then lead us to the prediction, the abundance PCA fitness. And so these are some of the graphs that show some of these relationships. We have the linear order, which is first order linear model here, which is the one of the higher performers. This is the predicted fitness on the y-axis, the observed fitness on the x-axis. So the first order linear model does a decent job of matching the observed fitness. The first order biophysical model it's roughly similar with respect to observed fitness versus predictive fitness. The first and second order biophysical model improves upon the first order biophysical model and the first order linear model. And then down here we have ddPCA training data, where we have these two models that are less predictive than these other ones. So this is where we get this so then we have the pairwise energetic couplings, which also improve genetic prediction. We have strong energetic couplings, which are due to physical contacts and backbone propagations. So finally, we talked about accurate multi-phenotype genetic prediction. And they say that the exceptionally large number of genetic backgrounds, which both single and double amino acid mutant effects were measured for the two related molecular phenotypes, this can serve as a rich source of data for biophysical modeling. So, we know that we can use deep learning methods, and that those are good, but they don't give a lot of biological context. But when we do this kind of method that they present here, it actually provides a very rich source of data for biophysical modeling, and we have a pipeline now. So firstly, considering only the abundance phenotype, we observed that an additive two-state thermodynamic model with unfolded and folded energetic states intermingled outperforms a linear model when evaluated on held-out variance. So they hold back some of the samples and they evaluate it. In order to attain similar predictive performance as the first-order biophysical model, requires including both second and third-order genetic interaction terms in the linear model. So these genetic interaction terms, as generic as they are, need to be included. This represents a massive increase in model complexity, however, so it may be, the gain that you get may not be worth it. This apparent greater complexity of inappropriate models that use many specific pairwise and higher-order genetic interaction terms to capture global nonlinearities in data, or this global epistasis, has been referred to as phantom epistasis. And so that's one aspect of this work, is that it can contribute to this sort of understanding. Next, extending previous work, we use a neural network implementation of a three-state equilibrium model with unfolded, folded, and bound energetic states to simultaneously infer the underlying causal free energy changes of both folding and binding. So the double delta GF and double delta GB, as well as folding and binding energetic couplings, which is the triple delta GF and the triple delta GB. This model fits the data extremely well, explaining virtually all of the fitness variance and the inferred folding and binding for energy changes. Those are first-order terms, and those are well correlated with those obtained previously using an independent shallow double mutant library. This is the first time, at least to their knowledge, that a large number of folding and binding energetic coupling terms have been measured for any protein. So they're actually able to do a job of measuring these with their parameters and getting new types of characterization. Okay, so this is the reference list, and I wanted to look at some of these references in terms of where they are and what they are. So there are a lot of nice references in here on epistasis and protein folding and other things in biology that are really kind of, I'm not familiar with as much as I should be. So we have a number of different things here. We have some papers on deep learning and some other papers. We have learning the pattern of epistasis linking genotype and phenotype in a protein. This is from Nature Communications in 2019. This is where they talk about this assumption of the model of genotype and then relating that to a phenotype. We also have papers down here on robust deep learning based protein sequence design using protein MPNN. So this is an example of a deep learning paper on protein sequence design. This was published in Science. There are also generative models of graph-based protein design, so advances in neural information processing systems. Minimal epistasis interpolation for sequence function relationships This is from Nature Communications in 2020. This also has to do with this sort of idea of epistasis and sequence function relationships. This is protein 3D structure computed from evolutionary sequence variation. This is from PLOS One in 2011. We also have mapping the energetic and allosteric landscapes of protein binding domains, and this is from Nature in 2022. Again, this is this idea of sort of where they're getting their parameters from. This paper here is the genetic landscape of a physical interaction. This is from eWife in 2018. That's probably interesting if you're interested in some of what they're doing here. And finally, we have the citation here, which is a review on causes and consequences of genetic interactions, or epistasis. This is the annual review in genomics, human genetics, and this is from 2019. So this is a nice reference on epistasis. Okay, the second topic I'd like to talk about are two papers on something called emergent chirality. And so we'll see what that means in a minute, but this is where we get chirality or handedness. So the first paper is Emergent Chirality and Active Solid Rotation of Pancreas Spheres. And this is a tissue type in pancreas. And what we're trying to do is we're trying to explain the chirality of active solid rotation and how that emerges. So the abstract of this reads, collective cell dynamics play a crucial role in many developmental and physiological contexts. While two-dimensional or 2D cell migration has been widely studied, how three-dimensional geometry and topology interplay with collective cell behavior to determine dynamics and functions remains an open question. So we understand 2D cell migration, and we talked about that in one of the papers earlier in the meeting, but this is three-dimensional geometry and topology that is on top of the cell migration model. So we don't really understand that very well. In this work, we elucidate the biophysical mechanism underlying rotation in spherical tissues, a phenomenon widely reported in both in vivo and in vitro. Using murine pancreas-derived organoids as a model system, we find that epithelial spheres exhibit persistent rotation, rotational axis drift, and rotation arrest. So there are these epithelial spheres that they've created from organoids, from pancreas-derived organoids. They exhibit these different aspects of rotation, and you can characterize all of these in this model. Using a 3D vertex model, we demonstrate how the interplay between traction force and polarity alignment could account for these distinct rotational dynamics. So this is again talking about the alignment of the tissue and the traction forces on the substrate. Furthermore, our analysis shows that the spherical tissue rotates as an active solid and exhibits spontaneously chiral symmetry breaking. So this is where, again, we have this chiral symmetry breaking in the model. Using a continuum model, we demonstrate how the types and location of topological defects in the polarity field underlie the symmetry breaking process. Altogether, our work shows that the tissue chirality can arise via topological defects and the pattern of cell traction forces. So this is where you want to know how tissue chirality arises. It's basically through topological defects. So topological defects lead to a symmetry breaking event, which leads to this chirality or this handedness. And so then you also have this pattern of cell traction forces, which allows for this topology to be formed and the defects to be formed on that substrate, with potential implications for left-right symmetry breaking processes in morphogenetic events. So this is, of course, a very general term, but in development, we have a lot of issues with symmetry breaking and with left-right symmetry breaking. So our heart is on one side of our body or another. We have differentiation between the different sides of the body, and this is symmetry breaking in development. So this just shows an example of the organoid derived tissue that we're using. So we have this cross section here. There's a lumen in the middle, and we have this layer of cells on the outside. It's in a matrigel matrix, so they're using this matrigel dome to house these organoids. So this is the substrate we're talking about. This is the azimuthal angle here. You can see then we have the sphere, as we can see here, and there's this rotational symmetry. And so the idea is that there are defects, random defects in the substrate, and that leads to symmetry breaking, which then leads to, in development, this sort of chirality. So you have this short-term state where you have rotation versus no rotation, and long-term behavior where you have rotation arrest and angular velocity axis drift. So you get these different rotational behaviors that you can observe, and this is the polar coordinate map of this type of short and short-term states and long-term behaviors. In this case, we have rotation around the axis. We can see angular velocities clustered at the pole. We have this case of no rotation where we have these these points that are more scattered. Rotation arrest where the points are scattered a little bit more, but where there's rotation, but it kind of stops. So there's rotation around the pole, and then when it arrests, it stops. And then you have this angular velocity axis drift, where the axis drifts off of the pole and goes to a different point. So this is where you get this asymmetry, this symmetry breaking, and you end up with a chirality. And so these points here are color-coded by their time. So you have from t0 to the end of the sequence. And in the first case of rotation, normal rotation, everything is basically in the same place. In this example of no rotation, you have these points at the beginning start kind of at the pole and then drift away, but there's no pattern to it. In rotation arrest, you get drifting away from the pole over time, and then angular velocity axis drift actually leads to this stable symmetry breaking. So you can see here where the axis changes measured over time, and you can see the differences. And so they actually talk about these two different phases of cell collective behavior, these solid and flowing regimes in an active vertex model. So this is where these various forms of dynamics appear within the same culture condition, suggesting that an underlying biophysical mechanism is responsible for the observed types of dynamics and transitions between them. To identify this mechanism, we developed a mechanical model of pancreas spheres using a 3D vertex model. So vertex models have been used to study the mechanical aspects of epithelial morphogenesis that regulate packing geometry, motility-driven rigidity transition, and developing tissues as these amorphous solids that exhibit yielding transition under shear. So you can use these kind of 3D vertex models for a number of applications. And again, we see our hexagonal arrangement here of our cells. We have these trijunctions here, and we have the polarity, which are different in these different cells. So we have these vertices for each these three cells. We have polarity driven by pressure here that are in different directions, which is kind of interesting. The traction force is moving in different directions here. So this is the shear force that's operating. This is C here, which shows the force fields. And again, in some of these examples, they're sort of aligned, and in some of these examples, they're non-aligned. So these last two examples are non-aligned. So this is where we have a solid regime. This is where we have a flowing regime. Okay, so that's the first paper. The next paper is Emergent Chirality and Current Generation. And this is another paper where they're looking at rotational motion. They're looking at emergent chirality and how currents are generated, electrical currents. So we investigated the phenomena of producing vibration-induced rotational motion in a cylinder filled with a chiral rods and being vibrated by an external drive. The arrangement of the rods develops chirality as a result of the interaction of gravity and steric hindrance, which is responsible for the induced motion. A two-rod arrangement is sufficient to generate a persistent motion. So this is where they're not using biological constituents, but they're using a physical model. A two-rod arrangement is sufficient to generate a persistent motion. The average angular velocity of the rods at long times varies non-monotonically with the packing fraction for a given drive strength. The precise nature of the variation depends on the details of the interaction between individual rods. Its general characteristics hold true for rods of various materials and geometries. A stochastic model based on the asymmetric simple exclusion principle or process helps in understanding the key features of our experiments. So here's where they do a very simple experiment. They have this simple experimental scenario where two pencils are placed in a container, as you see in A. Then in B, you have the schematic representation of A, where you have this angle at the bottom of the container to the top of the container for each pencil, and they're aligned opposite one another. And then 3 is a cylinder of earbuds, generates a doubly ruled hyperbolic surface. So you have these cotton swabs that are in this container, and when you have a lot of them, they kind of twist and, fitting them in. So they have this different geometry than the pencils. They're less rigid, and they have this, you have to squeeze them into the container. So they get this sort of curvature that you don't see in the pencils. Take note of the profile of the earbuds top ends, which are here. The rods on the inside are straighter than the ones on the outside. So in the inside of the massive cotton swabs, those are straighter, and you can see them kind of sitting upright. Whereas the ones on the edge are compressed downward and they're curved, and they have this, they're lower in this container. So take note of that. That's going to play a role in this emergent process. And then a typical snapshot of the system of rods being driven by a vibrational force. So they're being driven around. So you have this packing here. You already have an anisotropy, and then you have this driving force, which leads to this order that we are looking for. And so the component EH can be measured by the angle made by a rod with the wall of the container. A single toothpick and corresponding EH are highlighted. So this is the specific EH of a rod. It's now aligned from this. And so you have a bunch of rods that are aligned. So the rod packing increases with the number of rods. So you can see here that if you have 2 rods, they kind of can sit there at opposite ends. There isn't very much. They kind of jumble around in this thing, and there's no real order. The more rods you have, the thicker, the more dense the packing, the more order you get from this vibrational force, because they're interacting with each other and they're kind of moving around. So apart from the linear drift, there exist oscillations about the linear function corresponding to the rattling motion of the rods observed in experiments. For low densities, each rod can access a large section of the configuration space. So we know that for low densities, this rod can move around, and it can be configured in a number of different ways over the course of the shaking. On being driven by the shaker, the injected energy does not fully contribute to the global motion of the rods, but is used by individual rods to explore nearby configurations in its allowed configuration space. So this is where the shaking, if you have more rods in here and the packing is denser, then the shaking induces this, not only the constraint of the single rod to explore sort of a limited part of its configuration space, but all the other rods to explore configuration space. And in this collective exploration, they explore more of the configuration space, as opposed to just like one rod that's bouncing around and kind of hitting different parts of that configuration space at random. This leads to a noisy rattling motion of the rods at low densities, making the motion of an individual rod seem essentially random on short timescales. So if we did this experiment for 20 seconds, the rods would bounce around and maybe hit a sort of a random smattering of the configuration space. The rattling of the rods decreases as the number of rods in the container increases. since the available configuration space for each rod decreases. And so the system eventually becomes rigid. Okay, so this is where it explores its configuration space. Collectively, more of the configuration space is explored, and it ends up in a stable state that's rigid. Each rod is tilted at an angle with respect to the vertical along the wall of the container. We quantify this tilt as the tangential component EH of the axial vector. So this EH in the last figure was an example of one rod that's in a certain configuration. And so we can measure that as tangential component EH of the axial vector. So we can have an axial vector that describes the entire collective structure, but we can have individual rods that have a tangent to the axis. And so this map describes the free energy profile of this. And remember, we saw the example of the cotton swabs that had the crown of aligned rods, and then at the edge the rods were curved and downward, and this is kind of the energy profile of that. So it's replicating that pattern. And you can see that the red, which is the negative chirality, and black positive chirality markers indicate configurations that satisfy the condition D equals 2R, with D and R being the shortest distance at point of contact of two rods and the radius of the rods respectively. So for the contact of the two rods is equal to the radius, so it's basically the diameter of the rod. And so this is where, if you have more rods, you have you're exploring less of your configuration space. So this is a nice experiment that kind of relates to some of the things we've been talking about with respect to configuration space and packings. Thank you for paying attention. I hope you learned something.